Take a breath. Go ahead. Right now, breathe in, hold it for a moment. Feel it sitting there inside your chest. Now let it out. You just did the most complicated physics experiment you will ever perform, and you had absolutely no idea what happened. None. Zero. You think you do. You think air went in and air came out, and that is the whole story. But let me ask you something that should make you uncomfortable. What happened to the air while it was inside you? Not the general idea, not the children's textbook version where oxygen goes into your blood. I mean, what actually happened? Where did each molecule go? What did it collide with? What forces grabbed it? What changed about the air that came out compared to the air that went in? Because if you follow that breath molecule by molecule through the entire apparatus, if you really track it with the patience of a physicist, you do not end up feeling reassured. You end up genuinely alarmed. You end up realizing you are alive right now because of a process that has no idea you exist and no particular interest in keeping you around. Let me build this for you, one layer at a time, starting with something so simple it sounds insulting. The air around you right now, what is it? Most people, if you ask them, say oxygen. That is the default answer, oxygen. As if the atmosphere is a tank of the stuff and you pop open the valve and help yourself. But that is not what air is. Grab a fistful of air right in front of your face, an invisible handful. About 78% of what you are holding is nitrogen. 4/5 of it. Nitrogen. A gas your body cannot use, does not want, and must handle with every single breath. Another roughly 21% is oxygen. That is the good stuff. That is the fuel. The remaining 1% is argon, carbon dioxide, water vapor, and a few trace gases so scarce they barely matter. So already the situation is different from what you imagined. Every breath you take, you are mostly inhaling something completely useless to you. Your body hauls in this enormous load of nitrogen and does absolutely nothing with it. It is like ordering a package online and it arrives in a crate 20 times the size of the item. You rip through all that packing material to get to the one thing you need, and you do this 20,000 times a day. All right, down it goes. This mostly nitrogen cocktail with some oxygen dissolved in it, so to speak. And here is where the first real surprise lands. People picture the lungs as two balloons, big pink sacs. Inflate, deflate, done. But lungs are not balloons. They are one of the most elaborate branching structures in all of biology, and the reason for that branching is physics. Air enters through your nose or mouth and travels down the trachea. Your windpipe, it is a tube about the diameter of your thumb, reinforced with C-shaped rings of cartilage to keep it from collapsing under the negative pressure of inhalation. Air rushes down this tube, and then it forks. The trachea splits into two bronchi, one angling into each lung. These are still big tubes, but smaller than the trachea, and each of those splits again, and the daughter split, and the granddaughter split. This goes on for 23 generations of branching. 23. Starting from one tube, you end up with millions of tiny passages, like a tree turned upside down with its roots in your chest. Here is what this accomplishes physically. Each time a tube divides, the combined cross-sectional area of the two children is larger than the parent. Add up the areas generation after generation, and by the time you reach the smallest tubes, the terminal bronchioles, the total cross-sectional area has expanded enormously. The air that was rushing down the trachea at maybe a meter or two per second has slowed to a crawl, a gentle drift. This is critically important because of a concept called the Reynolds number. It tells you whether flow in a tube is turbulent, with eddies and swirls and chaos, or laminar, with smooth parallel layers gliding past each other. At the trachea, flow is somewhat turbulent, noisy, energetic, but by the time the air reaches the deep branches, it has slowed enough that the flow becomes laminar, and then in the final branches, the air is barely moving at all. Transport in those last centimeters is dominated not by bulk flow but by diffusion itself. The molecules are drifting to their destination under their own random thermal motion. The plumbing is designed so that the delivery method changes smoothly from convection to diffusion as you go deeper. That is elegant engineering. At the tip of each terminal bronchiole there is a cluster of tiny sacs. Picture a bunch of grapes hanging from a vine. These are the alveoli. Each one is roughly 1/3 of a millimeter across. A speck. You cannot see one with your naked eye, and you have around 300 million of them, in 2 organs that fit between your ribs. If you could unpeel all 300 million and spread them flat, the total surface area would be somewhere between 70 and 100 square meters. That is the size of a tennis court, give or take. A tennis court of membrane folded up inside your chest. Why so much surface? Because the fundamental rule of exchange between a gas and a liquid is this. The rate of transfer depends on the area of contact. Double the area, double the transfer rate. The lungs solve this problem with a ferocity that should impress any engineer. Instead of 2 big sacs with a modest surface area, you get 300 million tiny sacs with a colossal one, same volume, incomparably more surface. But here is where a nasty physics problem enters. All those tiny sacs are wet on the inside. They are lined with a thin film of fluid, and fluid has surface tension. If you have ever blown a soap bubble, you know the film tries to contract, tries to make the bubble smaller. Surface tension pulls inward. And Laplace's law says the pressure inside a bubble is proportional to the surface tension divided by the radius. Smaller bubble, higher internal pressure. Think about what this means for the alveoli. You have millions of tiny wet bubbles. The small ones have higher pressure inside them than the big ones. So air should flow from the small alveoli into the large alveoli. The small ones should collapse, the big ones should get bigger. A runaway catastrophe. You would end up with a few bloated air spaces and millions of collapsed ones. No gas exchange. The system should fail on the very first breath. Except it does not. Because your body produces a substance called surfactant. Specialized cells in the alveolar walls secrete this slippery mixture of phospholipids and proteins that coats the inner surface and reduces the surface tension by an enormous factor. Think of it as biological detergent. It breaks the grip of the fluid film on itself. Without surfactant, the Laplace instability wins, and the lungs cannot stay inflated. Babies born very prematurely, before about 34 weeks of gestation, often lack sufficient surfactant. This condition, called respiratory distress syndrome of the newborn, was one of the leading killers of premature infants for decades. It is not a problem of lung size or muscle weakness. It is a problem of physics, an unresolved equation. Modern medicine treats it by spraying artificial surfactant directly into the baby's lungs, and it works remarkably well because the problem was never biological complexity. It was Laplace's law. So, let me tally what we have needed so far before a single oxygen molecule has crossed the membrane. We needed branching geometry to maximize surface area. We needed fluid dynamics to slow the airflow from turbulent to diffusive. And we needed a chemical trick to defeat surface tension. 3 physics problems, 3 solutions, all just to get the air to the right place in the right condition. Now let us get to the main event. The air is sitting in the alveoli. On the other side of each alveolar wall, barely half a micrometer away, is blood. That wall is thinner than a soap film. You could stack 2,000 of them across a single millimeter, and the capillaries are so narrow that red blood cells have to squish and deform to slide through single file, pressing their membranes right up against the alveolar wall. Maximum intimacy between gas and blood. Minimum distance. Why does oxygen cross from the air into the blood? There is no pump, no valve, no little molecular hand reaching through to pull it across. The answer is diffusion. And if you understand diffusion, you understand the engine of your entire existence. Every oxygen molecule in the alveolar air is moving fast. At body temperature, oxygen molecules travel at roughly 400 to 500 meters per second between collisions. That is faster than a bullet from a pistol, but they are not going anywhere in particular. They are bouncing, crashing into nitrogen molecules, into other oxygen molecules, into the walls, changing direction randomly with each impact. Billions of collisions per second, complete chaos. No plan, no destination. On the air side, there are many oxygen molecules. On the blood side, far fewer, because the blood arriving at the lungs has already delivered most of its oxygen to hungry tissues all over the body. There is a concentration difference, a gradient, and because all those molecules are moving randomly, more of them will happen to bounce from the crowded side to the empty side than the other way. Not because they want to, not because they sense the gradient, simply because when more molecules start on one side, more random motions happen to carry molecules across in that direction. This is diffusion. It is one of the most important processes in all of nature, and it runs on nothing but thermal randomness. Let me give you a homely analogy. You are in a dark room at a party. One side of the room is packed with people, the other side is nearly empty. Everyone is wandering blind, bumping into each other, bouncing off walls. Over time, the room fills evenly, not because anyone planned it, because random motion plus a density difference equals net flow. That is all. That is the engine. And yet, for centuries, nobody believed this. Nobody believed that invisible particles could be crashing around at tremendous speed in ordinary air. The idea seemed absurd. Air felt calm, still, peaceful. It took a botanist looking through a microscope in 1827, Robert Brown, to notice that tiny pollen grains suspended in water were jittering around in a strange, erratic dance. He could not explain it. No one could. For decades. It was not until Einstein, in 1905, published a paper showing that this jittery motion was caused by invisible molecules of water constantly slamming into the pollen grains from random directions, that the reality of molecular motion became undeniable. The pollen grains were being battered by a ceaseless hail of molecular impacts. The jitter was the visible signature of the invisible chaos. That same chaos is what moves oxygen across your alveolar membrane. The same molecular pandemonium that jostles a pollen grain in a droplet of water is the force that keeps you alive. It is not gentle, it is not orderly. Each oxygen molecule is being hit, deflected, spun around, smashed into by its neighbors billions of times per second. The trajectory of any single molecule is completely unpredictable. A drunkard's walk in 3 dimensions at 500 meters per second. But out of that randomness, because the concentration is higher on one side of the membrane than the other, a net flow emerges, not from any force that aims it, from statistics alone. I want you to appreciate the numbers. In a single cubic centimeter of air, at normal conditions, there are roughly 2.7 times 10 to the 19th molecules. 27 billion. That is more molecules in a thimbleful of air than there are grains of sand on every beach on Earth. And every single one of them is moving independently, at speed, colliding and ricocheting off each other. The chaos is so thorough and so massive that the average behavior becomes perfectly predictable, the same way that if you flip a coin 1 billion times, you can predict with extreme precision that roughly half will be heads, even though you cannot predict any individual flip. That is why diffusion works so reliably. The individual molecules are doing absolutely nothing predictable, but the collective behavior, the net flow from high concentration to low, emerges from the numbers with the force of mathematical certainty. Your life depends on a statistical average, and the average is so good because the numbers are so large that it never fails, not once in your entire life. The quantitative version is Fick's law. It says the rate of diffusion across a barrier equals the diffusion coefficient of the gas, times the surface area, times the concentration difference, divided by the thickness of the barrier. Every variable matters. The lungs push each one to the extreme. Enormous surface area, steep concentration gradient, because the blood keeps flowing, carrying away oxygen, so the blood side stays hungry. Membrane, as thin as physics and biology can manage. Each parameter is not just good, it is pushed to the limit. And the speed is astonishing. A red blood cell passes along an alveolar capillary in about 3/4 of a second. That is its entire window. In that 3/4 of a second, the hemoglobin in that red blood cell goes from roughly 75% oxygen saturation to 97 or 98%, nearly fully loaded, in less time than it takes to snap your fingers, all from random molecular bouncing. At the same time, the reverse exchange is happening. Carbon dioxide, the waste product of cellular metabolism, is dissolved at high concentration in the venous blood. The alveolar air has very little carbon dioxide. So carbon dioxide diffuses from blood to air, crosses the same membrane, enters the alveolus, and gets exhaled on your next breath. 2 gases crossing in opposite directions at the same time at the same wall, driven by the same physics, no machinery required. Let me pause on that for a moment because I want you to feel the weight of it. The thing keeping you alive right now is not a pump, not a filter, not a mechanical device of any kind. It is random thermal motion acting across a concentration gradient on a surface thinner than cellophane. That is it. Every cell in your body depends on this every second, and it is nothing but statistics. Now remember our friend nitrogen. 78% of every breath. It dissolves in your blood a small amount, obeying Henry's law, which says the amount of gas that dissolves in a liquid is proportional to the partial pressure of that gas. At sea level, a manageable trace of nitrogen dissolves and sits quietly in your tissues. You never notice. But what happens if you increase the pressure? In the 1860s and 70s, something terrible was happening to workers in New York and St. Louis. They were building the great bridges, the Brooklyn Bridge, the Eads Bridge, and the foundations had to be sunk deep underwater in pressurized chambers called caissons. Workers would descend into these chambers where the air pressure was 2, 3, 4 times normal to keep the water out. They would dig and haul for hours. Then they would come back up to the surface, and some of them would collapse, doubled over in agony. Now joints screaming, some lost feeling in their legs. Some could not walk, some died. They called it caisson disease because nobody understood it. The workers who suffered the worst were those who came up the fastest. The ones who took their time seemed to do better, though nobody had a theory for why. What was happening was Henry's law running a vicious cycle. At the elevated pressures in the caisson, nitrogen was being forced into solution in the workers' blood and tissues. Their bodies were quietly absorbing enormous amounts of dissolved nitrogen into the fat, into the joints, into the spinal fluid, into the brain. As long as the pressure held, the nitrogen stayed dissolved and caused no mechanical damage, though at high enough pressures it acted as an intoxicant, making men sloppy and confused. The disaster struck on the way up. When pressure dropped rapidly, the dissolved nitrogen could not escape through the lungs fast enough. The blood became supersaturated. Tiny bubbles of nitrogen gas nucleated throughout the body. In the joints, these bubbles caused excruciating pain. In the blood vessels, they acted as embolisms, blocking flow. In the spinal cord, they compressed nerves, causing paralysis. In the brain, stroke-like damage. Once people finally understood the physics, the treatment was straightforward in concept, if difficult in practice. Recompress the patient, put them in a sealed chamber and bring the pressure back up so the bubbles dissolve again. Then bring the pressure down very slowly over hours, so the nitrogen can escape gradually through the lungs in an orderly fashion. You are literally rerunning the physics at the correct pace. Today we call it decompression sickness or the bends, and it remains a serious hazard for divers who ascend too quickly. It is nothing more than Henry's law and Dalton's law playing out in human tissue. The nitrogen that you thought was harmless, that silent 78% of every breath, is always ready to become lethal if you change the pressure wrong. Same gas, same body, different pressure, different outcome. Think of it this way. Open a bottle of soda. The cap comes off, the pressure drops, and dissolved carbon dioxide comes roaring out of solution as bubbles. You can see them forming, nucleating on the glass, rising to the surface. That is exactly what happens in the body of a diver who ascends too fast, except the bubbles are nitrogen, and they are forming not in a bottle, but in living tissue, in your joints, in your blood vessels, in your nervous system. Every open bottle of soda is a tiny, harmless demonstration of the same physics that can kill a diver. You have seen this happen 1,000 times on your kitchen counter without once connecting it to what happens inside a human body at depth. And here is what makes it particularly treacherous. The nitrogen dissolves into different tissues at different rates. Blood absorbs nitrogen quickly. Fat absorbs it slowly, but holds more of it. Your brain, which has a high fat content, accumulates nitrogen gradually during a long dive. Your joints and connective tissues take it up at yet another rate. This means that the decompression schedule is not one simple calculation. It depends on how long you were at depth, how deep you were, which tissues absorbed how much. The Navy and the diving community have developed elaborate decompression tables based on mathematical models of nitrogen uptake and release in different tissue types. All of this elaborate engineering exists because of one simple law: the amount of gas that dissolves in a liquid is proportional to the pressure of that gas above the liquid. Henry figured that out in the early 1800s. The rest is just working out the consequences. Go the other direction now. Higher. At roughly 5,500 meters, the air pressure is about half of what it is at sea level. Composition is the same, still 21% oxygen, but the partial pressure of oxygen is halved. The driving gradient that pushes oxygen across your alveolar membrane has been cut in half. Your body fights this. Immediately, breathing rate increases. Heart rate rises. Over days and weeks, your body produces more red blood cells to carry whatever oxygen it can extract. Your kidneys release a hormone called erythropoietin that stimulates the bone marrow. This is acclimatization. It works to a degree. People live permanently at 4,000 meters in the Andes, in Tibet, in Ethiopia. Their bodies have adapted over generations, with subtly different hemoglobin responses and higher red blood cell counts. But there is a ceiling. Above about 8,000 meters, the death zone, the partial pressure of oxygen, is so low that no amount of acclimatization suffices. Your hemoglobin cannot load, your tissues starve, your brain dims. Every minute at that altitude degrades you. Climbers on the summit of Everest without supplemental oxygen describe confusion, hallucination, an overwhelming desire to simply sit down and stop. That desire is oxygen deprivation slowly shutting down the brain. The gradient has collapsed below what the physics of diffusion can sustain. The same mechanism that runs effortlessly while you sleep at sea level simply folds when you change one number. The whole system, the branching, the surface area, the thin membrane, all of it optimized for sea level conditions. Shift the pressure significantly in either direction, and the physics turns against you. You live in a corridor of acceptable pressures, and the corridor is not wide. Now I need to tell you something about carbon dioxide that will change how you think about breathing. But first, consider what happens when you run, when you sprint or climb stairs or do anything that makes your muscles burn. Your cells are consuming oxygen much faster than at rest, maybe 10 or 15 times faster, and they are producing carbon dioxide at the same elevated rate. The blood returning to your lungs is darker, more depleted of oxygen, and more loaded with carbon dioxide than it was when you were sitting down. The gradients across the alveolar membrane steepen. Oxygen diffuses in faster, carbon dioxide diffuses out faster. Your breathing rate doubles, triples, may go up sixfold. Your heart pounds, more blood flows through the pulmonary capillaries per minute. Everything ramps up. And it works beautifully. Your body can increase its oxygen consumption from roughly 1/4 liter per minute at rest to 3, 4, even 5 liters per minute during hard exercise, depending on your fitness. That is a 20-fold increase. And the lungs handle it. The same membranes, the same alveoli, the same physics. How? Because the system at rest is not running at full capacity. There is enormous reserve. At rest, blood passing through the lung capillaries completes its oxygen loading in about 1/4 second. But it has 3/4 second of contact time available. There is a safety margin of about 1/2 second. During intense exercise, blood flows faster, contact time shrinks. But because the loading was mostly complete in that first 1/4 second anyway, the hemoglobin still gets adequately saturated even with the shortened window. The margin gets thinner, but it holds until it does not. Elite athletes pushing their absolute limits sometimes hit a point where the contact time becomes too short and the cardiac output too high for the lungs to fully oxygenate all the blood passing through. Arterial oxygen levels actually drop slightly during maximal exertion in some highly trained individuals. They have outrun their lungs. The heart is more trainable than the lungs. You can build a bigger engine, but the gas exchange surface does not grow to match. The physics sets a ceiling. People treat carbon dioxide as pure waste. You make it, you dump it, goodbye. But carbon dioxide is the master regulator of the entire respiratory system. When carbon dioxide dissolves in blood, it reacts with water. The product is carbonic acid. Carbonic acid releases hydrogen ions, which lower the pH of the blood, making it more acidic. Your brainstem monitors this pH with exquisite sensitivity. Normal arterial blood pH is about 7.4. If carbon dioxide rises, pH drops, and the brainstem ramps up your breathing to expel the excess. If carbon dioxide drops, pH rises, and breathing slows. Your breathing rate is not controlled by oxygen. It is controlled by carbon dioxide. The acid signal. The body is asking, is there too much carbon dioxide? Far more urgently than it asks, is there enough oxygen? There is a secondary oxygen sensor, the carotid bodies in your neck, but their signal is weaker and slower, and for the most part, carbon dioxide runs the show. This creates a trap that has killed swimmers for as long as people have been swimming. If you hyperventilate before a breath-hold dive, you blow off a large amount of carbon dioxide. Your blood pH rises. You have artificially lowered the carbon dioxide alarm threshold. When you then swim underwater, oxygen steadily depletes, but the urge to breathe does not arrive on schedule because the carbon dioxide signal has been suppressed. You can lose consciousness from oxygen deprivation before the carbon dioxide alarm fires. Shallow water blackout. It claims experienced, healthy, strong swimmers because they manipulated the control signal without understanding the system it controls. Two gases, moving in opposite directions, one secretly running the entire feedback loop. Remove that signal and the machine fails silently. No siren, No backup alarm strong enough. Just a slow fade to black. Let me take you deeper still, into the alveolus itself. The air sitting in an alveolus at any given moment is not the air you just inhaled. It has been altered. Oxygen has been removed. Carbon dioxide has been added. Water vapor has entered from the wet lung surface. It is warmer, wetter, and differently composed than outside air. And crucially, you never fully empty your lungs. Even after the hardest exhalation you can manage, about 1¼ liters of air remains trapped. The residual volume, so each fresh breath does not flush the system. It dilutes the old mix. ½ liter of new air blending into several liters of old, like adding clean water to a tank, cup by cup. This sounds inefficient, but it is actually vital. It buffers the alveolar gas composition, so the blood encounters a steady environment, not a wildly fluctuating one. Hemoglobin works best when the gradient is stable, and hemoglobin deserves its own segment because this molecule is a miniature marvel of physics. Hemoglobin sits inside red blood cells. Each molecule has 4 heme groups, each capable of binding one oxygen molecule. But the binding is not independent. When the first oxygen attaches, the hemoglobin changes shape. This conformational shift makes it easier for the second oxygen to bind, and the second makes the third easier, and the third the fourth. Cooperative binding. The molecule accelerates its own loading. In reverse, when one oxygen releases in a low oxygen environment, the others become easier to release. The molecule accelerates its own unloading. Plot oxygen partial pressure against hemoglobin saturation, and the result is an S-shaped curve. At the high partial pressures in the lungs, hemoglobin is nearly saturated, 97 or 98%. In the tissues where oxygen has been consumed and partial pressure is low, the curve drops steeply. Hemoglobin dumps its cargo right where it is needed. It is not a passive bucket, it is a responsive delivery system governed entirely by local conditions. Carbon dioxide makes it smarter. In tissues doing hard work, carbon dioxide levels are high. High carbon dioxide shifts the binding curve, so hemoglobin releases oxygen more easily. This is the Bohr effect. The tissues that need the most oxygen, the ones burning the most fuel and producing the most carbon dioxide, are exactly the ones where hemoglobin loosens its grip the most. No signal from the brain required, no hormone, just local chemistry pushing a protein into a different conformation. The physics does the thinking. Acidity doubles down on this. More carbon dioxide means more carbonic acid, means lower pH. And lower pH independently shifts the curve. The same